Okay, before we begin, are there any questions? Can somebody confirm that you can hear me?
We can hear you.
Thank you. I did get some grading done, so take a look at the well, your grades, I guess. Let me stop sharing the screen. I'll call up the grade book. I think I'm not sharing the screen. I think lab 0, lab 1, and lab 2 are now graded. Let me double check that though. For the first time, not for the second time. I'm not sure when I'll get to grading the second time. Shrink that. Yeah, the Plagy quiz is graded. The extra credit picture assignment is graded. The extra credit study sheet is graded. Extra credit study 2 is graded. Worksheet for lecture 1 and worksheet for lecture 2 is graded. The quiz 1C is graded. I think one of the extra credit quizzes I got graded, but let me check on that. I'm not seeing that now. And then, yes, the first grading of lab 0, lab 1, and lab 2 has been done. If you do not have 100% on lab 2, you can make corrections by this Saturday. Let me check on that to make sure. Yeah, January 27th. Started grading practice quiz 00A, but I have not gotten very far. And practice quiz 00B is not graded. So if you see a 0, it's because I haven't gone in there and checked for the correct answers. But it has as its value is 0 because it's extra credit point. So any points you get will be on top of the total points for the term. And I should really move these away from here, but I'm going to leave them there. But that needs to be moved. All right. Any question about any of that? Now, when I move things in the grade book, I don't know if that moves anything on your screen, but I'm moving the drop quiz right now, moving it to the quizzes, because it should be after the quizzes. And let me state, since I'm talking about the quizzes now, quiz 5C says it is out of 0.24 points. That's because if you were to add all of the quizzes together in the grade book, that would be more points for the quizzes than is possible for the course because you're gonna drop one of the quizzes. Quiz 5C will be worth the full points, which is 75 points, but I guess I should be stating this in the lecture, so I don't need to state it 3 times. Anyways, quiz 5C I will go in and manually change the grade from out of 0.24 to out of 75. And it'll be in the column. There's a column quiz 5C with a capital Q, and then a column quiz 5C with a lowercase q. I'm sorry, it's kind of confusing, but your points will be added to quiz 5C. So the total point for quiz 5C will be 75 points. And then on the drop quiz column, I'll drop your lowest quiz score. Any question about that? Anyways, that will make the total quiz points equal to what I have in the syllabus. And I don't remember how many it is, but you can look it up in the syllabus. The point is you get to drop one of the quizzes. And so the grade book has to be less than all of the quizzes. So that's why quiz 5C in that item, why it's out of 0.24 points. And that's to make it close to what it really should be, so that your grade will not be inflated. Because if you were to take all 5 of the quizzes and they'd all be 75 points and one of them wouldn't count, then your grade will be inflated in the grade book. And I'm trying to keep that from happening. Any question about any of that? All right, let's get out of the grade book and then I'll start the lesson. Let me share my screen again. Sorry, it's a share. It's brand new and I'm a little bit thrown by that. All right. I guess I'll leave that and open another window. So today is January 25th, week 3, and we should be studying lab 4 stains. Any question about what we're going to do? If not, let me go to lab 4. Lab 4, staining techniques. We're going to talk about the simple stain, the differential stain, and special stains. So read the textbook on size, shape, and arrangement of cells, page 73 to 75. I'll read about bacterial cell walls, read about the simple stain, read about the differential stain, and there's 2 differential stains we're going to talk about in this lab, the Gram stain and the acid-fast stain. And then lastly, read about special stains. We're going to discuss 3 special stains in this lab. The negative stain for staining for capsules, also called the capsule stain, the endospore stain, and then the flagella stain. There are some supplemental materials in this lab, including video clips, and there's a virtual lab in this lab. Your learning objectives for this lab: 1, be able to prepare a bacterial smear for staining. And let me emphasize that when you make a bacterial smear, you are not doing any staining. The bacterial smear is something you do to the slide before you do the staining. 2, understand the principles and the procedures used when performing the simple stain, the differential stain, and a special staining technique. 3, interpret the results for a simple stain, a differential stain, and special stains. And 4, be able to define these terms: simple stain, differential stain, special stain. What's the coccus? What's the bacillus? What's the spirillum? What is peptidoglycan? What's the gram-positive cell? What's the gram-negative cell? What is the acid-fast stain? What is a primary stain? What is a mordant? What is a decolorizer? what is a counterstain, what is a capsule, what is an endospore, and what is a flagella. When we look at bacteria under the microscope, they are essentially transparent. Therefore, to see them, at least see them well, it is necessary to stain them before they can be observed. When preparing to stain the cells, you do that on a bacterial smear. How you make a bacterial smear is you add a drop of sterile water to a clean slide. Then you add the bacteria into the water and you mix the bacteria into the drop of water. And then you smear the drop of water all over the surface of the slide. That will, for one thing, besides spread out the cells, it will also spread out the water so it'll dry quicker. You then allow the slide to air dry, and then you heat fix the cells by gently heating them. The air drying and the heat fixing are important so that the cells will not be washed off the slide during the staining procedure. If you do not air dry and heat fix the cells, they will wash off. A simple staining technique is one that uses one dye to stain a bacterial cell. You can determine the shape and arrangement of the cells, but they will all stain the same because you're only using one dye. A differential staining technique usually uses two or more dyes to provide additional information. And then the special stains are used to dye particular bacterial structures in the cell that you want to look at, such as the capsule, the endospore, or the flagella. When we're looking at bacteria under the microscope, they will take on a shape as well as a cellular growth arrangement, which is sometimes just called a growth arrangement. Or what is it? Cellular arrangement. Sorry, cellular arrangement. You can have cocci cells. They are spheres. You can have rod-shaped cells, and we call those bacilli or rods. You can have spirilla, and those are S-shaped cells, or if they have many spirals, we call them spirochetes, and that's the same as a spirilla, spirochete and a spiral. There are a few species of bacteria where the shape is pleomorphic, so the shapes will be differing between different cells, and this isn't the best picture of a pleomorphic cell. But you can see some differences here. That sort of looks like a footprint. That looks like almost like a spiral. That one looks sort of like a rod, although it's tapered. And that one's sort of a bent rod. That one's sort of a bent rod too. And then there are some cells which are in between a bacilli and a cocci, and those we call coccobacilli. There's three basic shapes that most bacteria take are the cocci, the bacilli, and the spirals or the spirilla. Most of the other shapes are some combination of these three, like the coccobacilli is a combination of the cocci and the bacilli. Any question about any of that? We're seeing a few of the growth arrangements. I think I'll talk about that in a later lab, but you can have a single cocci, diplococci, and that's two cocci stuck together. You can have tetrads, that's four cells stuck together. You can have staphylococci, and this is a cluster of cells stuck together. And then you can have cocci in a string, and these we call streptococci. Oh,
Dr. Volpe,
yeah.
with cocci, would you ever see them clumped any closer together, or will they always just be in clumps of 4 spaced out pretty nicely?
No, sometimes this cocci can be on top of each other.
They can be on
Okay,
top.
it just depends how the bacterial smear was made. If you have only a few cells, then you'll have a single layer of cells, and the cells will tend to be spaced out. So if you have tetrads, they would tend to be spaced out. But if you have cells on top of each other, you would have a hard time even seeing the tetrads because there'd be so many cells on top of each other.
Okay,
Okay.
thank you.
All right, let's talk about the differential stain. The most important differential stain and the most commonly used differential stain in microbiology is the Gram stain. It was invented by Hans Christian Gram, a doctor, in 1884, who devised it as a differential stain to be able to detect the bacteria that were causing an infection in a patient. And so when you spell out the Gram stain, it should have a capital G because this name is named after Dr. Gram, and we always capitalize somebody's name. Any question about any of that? Now, with that stated, Dr. Gram used it to differentiate between the bacteria cells, which stained dark purple in his samples, and the patient's own cells, which stained pink in his stain, or specimen, whatever you call it. Microbiologists started using the Gram stain, and they discovered that it was differential between different species of bacteria, that it could distinguish between different types of bacteria according to their ability to retain the crystal violet. Crystal violet is the first stain used in the Gram stain, and it's the primary dye. Bacteria that can retain the crystal violet in the Gram stain are classified as Gram positive, and they will appear purple at the end of the Gram stain. The bacteria that do not retain the crystal violet are referred to as Gram negative cells, and at the end of the Gram stain, they will appear pink. And the point is that the Gram stain can differentiate between different species of bacteria, between the Gram positive cells and the Gram negative. So it's a differential stain, as Dr. Gram intended it to be, but he didn't intend it to differentiate between bacteria. Any question about any of that? All right, here we're looking at a mixture of different bacteria cells, and you can see it was stained by the Gram stain. You can see a Gram negative bacillus right there, and that's a Gram positive bacillus right there and there and there and there. That's a Gram negative spirillum right there. There's a Gram positive bacillus, and there's some Gram positive cocci right there. Any question about any of that? All right, the Gram stain stains bacteria differentially based upon their cell wall composition. The Gram positive bacteria has a very thick peptidoglycan layer in the cell wall, while the Gram negative bacteria have a thinner peptidoglycan layer in their cell wall. And so the difference is just really the thickness and the peptidoglycan content of the cell wall. For the gram-positive cell, I wonder why that was cut. I'm sure that's bacteria or bacterium, and it seems to be cut. Anyways, the gram-positive bacteria is shown here. There's the cell membrane, and it has a very thick layer of peptidoglycan. Let me really blow this really high. And if I blow it up really high, you can see some of the layers in the peptidoglycan, maybe best over here. So there's 1, 2, 3, 4, 5, 6. Just in this region here, you can see 6 layers of peptidoglycan. The point is that the gram-positive cells have around, I used to know this now, is it around 100 or greater, or is it around 30 or greater? Oh, geez, I forgot that. I'll have to look that up again. It has hundreds of layers of peptidoglycan, one layer on top of the next, okay? And the point is, it's just many layers of peptidoglycan. It may only be 30, I don't remember, and I should remember that, but I don't remember. I do remember the gram-negative cells. Let's go over here. Here you see the gram-negative cell. There's the cell membrane. Here is the cell wall. This layer here of peptidoglycan. And then this lipid bilayer outside the peptidoglycan is the gram-negative cell wall. You can see some layers of peptidoglycan in here. I'm not sure which one is the best. Now, why did that switch? When I was shrinking it, it blew up. Maybe about right there. The point is there are between about 3 layers of peptidoglycan and 20 or so layers of peptidoglycan, but there are many. If you look at your textbook, it will show the peptidoglycan layers of the gram-negative cell is just one layer, and that's not right. There's several layers, but it's many fewer than the gram-positive cells. Any question about any of that? All right, after bacteria have been stained with crystal violet in the gram stain, we then stain the cells with gram iodine. Iodine is not a stain, it is a mordant. A mordant is something that chemically combines with the first stain, in this case crystal violet, and then forms a crystal violet iodine complex. The crystal violet iodine complex is a larger molecule than the crystal violet or the iodine alone. Next, we apply alcohol acetone as a decolorizing agent, and you remove the crystal violet from the gram-negative cells simply because it has a thinner peptidoglycan layer, and so the crystal violet washes out of the gram-negative cells. However, the gram-positive cells have a thicker peptidoglycan layer, and the crystal violet iodine complex does not wash out of the gram-positive cells. The gram-negative cells are then clear because there's no crystal violet in them. The gram-positive cells remain being purple in color. You then apply the counterstain, which is safranin in this case, to dye the gram-negative cells pink. And that's why the gram-negative cells are pinkish in color and the gram-positive cells are purple in color after the gram stain. Any question about the gram stain? All right, let me state that there are some cells which do not stain well in the gram stain. This is mostly the species that belong to the family Mycobacteria. The mycobacteria have a lot of macolic acid in their cell wall. And macolic acid is a lipid, and that lipid prevents water-soluble things from getting into the cell. The lipid also helps keep the cells alive because it doesn't allow water to leave the cell. The point is that the gram stain is it uses water to bring the stains to the cells. The stains typically do stain gram-positive, the mycobacteria, but they stain lighter than other bacteria because the crystal violet doesn't easily get inside the macolic acid of their cell wall. Sometimes when you're staining the mycobacteria, they will take on the appearance of a gram variable cells. And you can see gram variables with other species, particularly if the gram stain is done incorrectly. Now, all the times when I've ever seen the students show me a slide that look like this, where they have one species of bacteria in it, and then the cells show gram variable, meaning some are gram negative and others are gram positive. I asked the students, how old were these cells? And they told me, and it's always mycobacteria, by the way. And they told me, oh, these cells are at least 2 weeks old, maybe older. And I tell them, well, you know, with the gram stain, you should only apply the gram stain to cells that are 2 days old or younger. And if you use older cells, the gram stain may not stain correctly. And in this case, the reason why you're seeing the gram variable is because the cells are much older than 2 days old. What do you notice about these two cells, the dark purple gram positive staining cells, and this cell here, maybe that cell there, and let's say that cell there or that cell there. And it's kind of hard to go, so I'll go there. We'll say that one. The point of this one, which is, I'm going to say, is to the left of the pointer here. And then this one here, which is also to the left. What do you notice between these 3 cells compared to these 2 cells?
They're smaller.
Yeah, they are. What else do you notice? Meaning smaller is not what I was looking for.
Well, they're in between, so they're not pink or purple. Their color is different.
Their color is pink.
Is it the shape? They're kind of
Yeah, the
different.
shape is a little different. You'll notice that this cell looks broken. That cell, I'm not sure what's going on there. It's darker on the bottom, perhaps broken, and then lighter on the top. And this cell here, which is really hard to talk about, we'll say that cell there. That cell there is very light. All 3 of these cells sort of look like a ghost of a cell. Maybe if I shrink it down, that'll be more apparent. Nah, not really. I just call them, they look like a ghost of the cell. And the reason is these cells are probably dead. That's why that cell is breaking apart. And another thing you can notice is they don't have a very distinct boundary between the cell and the background of the cell. Whereas these cells, especially this one there and that one there, and that one there and that one there, they're very darkly staining and the boundary is very distinct. These are probably viable cells. Why they're so darkly staining and why their boundary is so distinct. Whereas, like I said, that one, you can easily see that cell is broken in two. The point is you've got old cells here, and old cells don't stain well with the gram stain. The gram stain can give false results if you have old cells in the slide preparation. And so all of these gram variable cells are probably cells that are either dead or dying, and they're not holding on to the crystal violet because the cell wall is deteriorating. Whereas the viable cells have an intact cell wall and they're holding on to the crystal violet. Any question about any of that? All right. So some tips to prevent getting gram variable stain of the bacteria. First, avoid using old cells. The cells should only be 2 days old or younger. Prepare a thin smear. If you have a very thick smear, it'll be difficult getting the dye into the lower layer of cells. And then if you do get the dye into the lower layers of cells, and then you use the decolorizing step, it'll be hard to wash out the crystal violet from the lowest level of cells. Heat fix the bacteria onto the slide only after the smear is air dried. If you heat fix the bacteria before it's air dried, the water will evaporate from the cell and that will damage the cell wall. And then the cells will not stain correctly with the gram stain. And then avoid overheating the cells during heat fixation. Excess heat also disrupts the cell walls. Use fresh reagents. And then this is an important one. Do not over decolorize or under decolorize. Because if you over decolorize, you will pull out the crystal violet from the gram positive cells. And if you under decolorize, you will not pull out the crystal violet from the gram negative cells. And we'll talk about the gram stain in a little bit. I'll show you how to make sure you decolorize for the right amount of time. All right, any question about the gram stain? So let's talk about the next differential stain we're going to talk about. We only talk about two, and that's the acid-fast stain. Some bacteria, mainly the mycobacteria, but also the family Nocardia, have a waxy substance called mycolic acid in their cell walls. These bacteria resist the acid alcohol decolorization in the acid-fast stain. And these cells, we call them acid-fast bacteria or acid-fast positive bacteria. Most bacteria, meaning essentially every family of bacteria that's not mycobacteria or nocardia, are acid-fast negative bacteria. And they will lose the acid-fast stain during the decolorizing step. In the acid-fast stain, you initially stain all cells with the primary stain, carbol fuchsin, and it's a cherry red in color. The cells are then decolorized with an acid alcohol decolorizer, and that removes the carbol fuchsin from the acid-fast negative cells. You then apply either methylene blue or brilliant green, or sometimes crystal violet, as the counter stain. And that will stain the cells that have been decolorized by the decolorizing step. So at the end of the staining process, the acid-fast cells will retain the red color, and the acid-fast positive cells, and then the acid-fast negative cells will lose the red color, and they'll pick up the counter stain. And the counter stain will be either blue or green, depending on what counter stain was applied. Any question about that? So when we look at a mixture of bacteria, the acid-fast positive bacteria will appear pinkish or reddish with the acid-fast stain, and then the acid-fast negative bacteria will appear greenish or bluish. And they'll be blue if it's methylene blue that's used, and they'll be green if it's brilliant green. Is that the one? Yeah, brilliant green. Any question about any of that? All right, let's talk about the special stains. There are 3 special stains we're going to talk about in this lab. The first is called the negative stain, which uses a negative stain to stain the capsule. You can also call this the capsule stain. The capsule stain is a stain that shows the capsule. Some bacteria produce a substance that we call the glycocalyx, which is secreted by the cell and exterior to the cell wall. If the glycocalyx is firmly attached to the cell wall, and you can see it in the light microscope, we call it a capsule. So if the glycocalyx is firmly attached to the cell wall and you can see it in the light microscope, we call it a capsule. If, on the other hand, and I don't think this lab talks about it, I'll talk about it later. If the glycocalyx is not firmly attached to the cell wall and you cannot see it in the light microscope, we call it a slime layer. The reason why you can't see it in the light microscope is because the slime layer is mostly water. But it is a glycocalyx, something secreted by the cell and exterior to the cell wall. You can see the slime layer with the electron microscope, but not with the light microscope. Productions of capsules by bacteria can be associated with an organism's virulence or its pathogenicity. And this is because the capsule helps protect the bacteria from being phagocytized by white blood cells. And we'll talk a little bit about that later. White blood cells can phagocytize bacteria. Let's eat them. And if the bacteria has a capsule, then the white blood cells cannot phagocytize them. If you look at the bacteria colonies on an agar plate, you'll note that if the bacteria produces a capsule, the colonies have this mucoid-like appearance. Capsules are not easily observed with conventional staining methods. For example, like the gram stain, the different steps of the gram stain tend to wash off the capsule from the cell. And therefore, we have special stains to stain or make the capsule appear. The negative stain is the only one I'm going to talk about. It uses a negative stain like India ink or nigrosin. A negative stain is one that stains the background of the cell, as well as staining the, I think I got a picture of it down here. As well as staining the, I don't have a good picture of it, sorry. As well as staining the cell itself. Now this, weird to me, doesn't look like a negative stain. This looks like a capsule stain. That's not terribly important at the moment. Let me see if I can find my picture. And I should put this in this lab. Either chapter 3 or chapter 4. I think this is the one. Our place. Because I'm not finding it here. Yeah. There it is. This is a negative stain. India ink primarily stains the background of the slide, why the background is so dark. However, India ink does lightly stain the cells as well, and they look kind of like a gray color because they're lighter than the background. You'll note that the negative stain does not stain the capsule. And the reason why, come on, mouse, the reason why these cells have a clear region around them is these cells have a capsule. And the negative stain does not stain the capsule, but stains the background of the cell slide, and it lightly stains the cells. The capsule stain, now let me take this picture. Oop, what happened here? Oh, there we go. Let me take that picture and move that into the lab. Move it over. Yeah, I'll try and shrink that. Come on. Ah, can't move it over. Ah, all right. I should do this later so you guys don't have to be watching me do it. Let me shrink that one. A little smaller. Come on, about there. Oh, got the wrong one. There, I got it. Um, all right, let's talk about the next special stain, the endospore stain. Endospores are metabolically inactive, hardy structures that are produced by some bacteria as a defense strategy so that the cells can survive harsh or unfavorable environmental conditions.
Dr. Volpe,
Yeah.
you're not sharing anything right now?
Oh, thank you.
You're welcome.
Why did I stop the share? Where is my Zoom? There we go. So you didn't get to see what I was doing. Well, that's good. What I did was I just put these in the lab module. So I got that one, which is both a negative stain and this one. Let me shrink that a little. There we go. Boy, that's taking a real long time to run. There we go. Let's delete that too. Not sure why they got so much space there. Anyways, endospores are things that allow the cell to live in a harsh environment. And then when the environment becomes favorable, the endospores germinate and the bacteria return to their normal. Sorry, not sure why that came up. The bacteria return to the normal metabolically active vegetative state. So the bacteria cell normally will be in the vegetative state, and that'll be a metabolically active cell. It'll be growing and reproducing. When it's in its endospore, it's sort of like in hibernation, and it has this thick, tough outer coat around it too. And the cells are essentially metabolically inactive. They're not totally inactive, but they're almost totally inactive. And the endospores can live for thousands of years. They have endospores they took out of Egyptian mummies. And if I remember correctly, that's something like 8,000 years old. And they germinated endospores that came out of cores from Antarctica, meaning they took a core of ice sample going down, I don't know, thousands of feet, and then pulled out the core, and they found endospores in the ice from something like 40,000 years ago. I think it might be 40,000 years ago. It might be 40 million, but I think it's 40,000 or maybe even 400,000. And they were able to get them to germinate. Most species that make endospores belong to two families, the genera or family Bacillus and the family Clostridium. I think there is one other family, but it only has one member that makes an endospore. All right, this table is showing you some of the endospore forming gram-positive bacilli, giving the bacteria species on the left and the clinical significance on the right, like Bacillus anthracis is the bacteria that causes anthrax. You'll need this table to answer some of the questions in the lab module. And why did that get pulled down? Let me see if I can bring this up. For some reason, I got a whole bunch of space in here. And normally it's not a problem, but that split the table there, so. All right. Oop. Having trouble saving this for some reason. I might have to close this down and restart it. It looks like it's gotten stuck. Oh, there it goes. Not sure what the problem is. All right. The Schaefer-Fulton stain is also called the endospore stain. It uses malachite green as the primary stain, and it stains all cells, including the endospores, green. Heat is applied as the mordant that helps the malachite green get inside the endospore. The cells are then decolorized simply with water, and water removes the malachite green from the vegetative cells, but it does not remove the malachite green from the endospores. Safranin is then applied as the counterstain, so any cells that are clear from the decolorizing step will then pick up the safranin. This will make the vegetative cells appear pink, and the endospores will be a green color. And this is the endospore stain, also called the Schaefer-Fulton stain. I'll blow this up a little. The pinkish cells are the parent cells that make the endospores, and they are vegetative cells. The green bodies by themselves are endospores that have come out of the parent cell. And that probably happened because the parent cell died. And then right here, we see an endospore being made inside of a cell. And you can see there and there, I'll talk about this one because that is another cell there. And that one has the arrow kind of on top of the cell. You can see that on top of the top portion of the cell, it's pink because that's the parent cell. And if you look, the endospore is totally inside the parent cell because at the bottom of the cell, there's a slight pink region. Any question about any of that? So endospores are made inside the bacteria. And then when the parent cell dies, that will release the endospore. All right, let's talk about the third special stain. And the last special stain we're going to talk about is the flagella stain. A flagella is a structure that bacteria have for locomotion. So if the bacteria have motility, meaning the bacteria can swim, it's because they have at least one flagellum, if not several flagella. Okay? And flagella is the only structure that bacteria have to allow the bacteria to swim through a liquid media. Flagella are composed of flagellin, that is a protein. You can talk about the distribution of flagella on the bacteria cell. You can have a monotrichous flagella, and that's one flagella at one end. You can have a lophotrichous flagella, and that's several flagella at one end. You can have an amphitrichous flagella, and that's several flagella on both ends. And then you can have a peritrichous flagella arrangement, and here the flagella will be all over the cell, coming out all over the cell. You do not need to know the different flagella arrangements. I will never quiz you on it, but you must use this figure to answer one of the questions in the lab module, the lab exercise. Because flagella are too thin to be seen using standard staining techniques, we have a special stain that allows the flagella to be viewed under the light microscope. Usually the flagella stain, what it does is it coats the flagella many times, meaning it puts down one layer, then puts down another layer and another layer and another layer, and you coat the flagella many times, and then the flagella can then be viewed and seen under the light microscope. Here we have an example of the flagella stain. Let me see if I can blow this up. You can see that this cell has many flagella on it, but that cell has fewer. That cell does look like it's peritrichous though, because the flagella are all over the cell. But this cell here is lophotrichous because it only has flagella on one side of the cell. But there's more than one. And that cell, it's hard to see, but that cell and maybe that cell is has only one flagella. The reason why the cells have a different flagella arrangement is not because they're different species. These are all species, the same species, meaning the same species was grown up, and then we made the flagella stain, and then some of them had many flagella, some of them had fewer flagella, some of them only flagella on one end of the cell, and then some, like maybe that one. Trying to see another example. I thought there were a couple good examples, but I'm not seeing them now. Maybe that one only has one flagella. And if you look real hard, that one doesn't look like it has any flagella. This one isn't, we're not seeing all of the sides, but it doesn't look like it has any either. That one may not have any flagella. And the point is, you'll note that the cells have more than one arrangement of flagella. The reason for this is because when the cell first divides into two cells, we'll initially have a cell that has no flagella. And then it'll start making flagella. And at the very first, it'll only have one flagella. And then it'll have flagella all on one end. So sort of like this cell, more than one, but all on one end. And I don't see any cells at the top that have flagella at both ends, but then that'll happen. The flagella will be a few at both ends. And then the cell will start putting out flagella all over the cell. And it'll be the peritrichous arrangement. So when you look at cells for the flagella arrangement, you look for the arrangement which is the highest order. And you say that this species of cell will produce that order, meaning in this case, the peritrichous flagella arrangement. And the other cells, if they're given more time, they will develop eventually into the peritrichous flagella arrangement. Any questions about any of that? All right, so know the terms of this lab. And then when we're doing this lab, remember everything's going to be done online, so you're not physically going to be performing any of these activities, but you must understand how to prepare a bacterial smear. You must understand the principles and procedures used for various staining techniques, and you must be able to interpret the results using a microscope. Now let's go to the exercises. Oh, come on. So in procedure 1, you're going to be preparing a smear for staining. We're not going to be doing the staining because that's not making a bacterial smear. We have a little video for you to watch on preparing and making a bacterial smear. And then after it's air dried, we have a little video on heat fixing the slide. And that would be the end of making the bacterial smear. How you make a bacterial smear, I've already mentioned it. You add a drop of water to the slide. You add the bacteria into the drop of water, and you add very small amount of bacteria because you only want the bacteria to be a single cell layer thick. You then allow the slide, you then spread the slide. You know, that's missing here. Then spread the mixture of cells and water all over the slide. Allow the slide to air dry. Heat fix the cells. This will stick, the air drying and the heat fixing will stick the bacteria to the slide, so the bacteria will not wash off. Let the slide cool, and now we're done making the smear. The smear can now be stained, but the staining is not making the smear. All right, and then answer this question for the worksheet. What are 4 common errors that students make when preparing and making a bacterial smear? So list 1, 2, 3, 4 different things that can be done wrong. Procedure 2 will be doing a simple stain. We have a video clip to watch on making a simple stain. You can review the steps of making a simple stain. You usually just add 1 dye to cells that have been put on a slide in a bacterial smear. You then remove the dye and rinse it, and then blot it dry with bibulous paper, and then you look at it under the microscope. All the cells will be the same color because this is just a simple stain. Here we have Staphylococcus aureus and E. coli, and then answer these questions. What's the cellular morphology, the shape? I don't want you to put the cellular growth arrangement. but the shape of 1, E. coli, and 2, Staphylococcus aureus. And let me warn you that E. coli is not a spiral. It looks like a spiral right there because 2 cells are hooked together. That might even be 3 cells. And there, right there, that looks like a spiral too, but that's 2 cells. And there you can easily see 2 cells, 1 there, and then 1 there. And then right there is 2 cells stuck together. Right here is actually, well, these are 2 cells stuck together, and then these right here, I think that's 3 cells stuck together. Right there is 2 cells stuck together. And that too is 2 cells stuck together. Sorry that this preparation was so poor. There's a lot of cells that are stuck together in this preparation. Any question about any of that?
Dr. Volpe, I've been looking at that specimen slide for a while now, trying to figure out bacilli. They look to be wider than those are on that slide.
What are you asking?
Well, I'm asking bacilli. They look to be shorter and wider, but I can't, to your point, like the slide quality is not that great. So I'm having a hard time determining if it's the
Okay.
shorter.
Bacilli are not always short and wide. Sometimes they're long and skinny.
Okay.
Okay. The bacilli can have various shapes.
Okay.
I haven't talked about that, but we will in a later lab. Let me go ahead and just show you the basic shape of a bacilli. I'm not going to do this very good because I can't do it. Can't draw in a straight line. Is a rectangle like that. However, the ends could be blunt like this, but they could be rounded like that. And they can even be kind of pointed. This is going to be hard to draw. Sorry, I'm not a good artist. And I'm doing this with a mouse and it's just not doing. So the bacilli can take on a different shape.
Okay.
Because of
That's
the
helpful.
way the edge is.
Thank you.
So it could be blunt edge, it could be rounded edge, and then it could be pointed edge. Okay?
That's helpful. Thank you.
All right. All right, and then question B, what is that? Procedure 2, number 3A, and then number 3B is what is the total magnification? Procedure 3 is the gram stain. So view the following video on performing the gram stain. And then we have a gram stain in a virtual lab. So open this virtual lab, and this may take a little while to download. Let me turn my volume down because this is a little loud. And you can go through the entire
Hi, lab
stain.
assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria.
I'm just going to quickly move through it to get to the part where the gram stain is, but you can go through the, whatever this thing is, the program, okay? So I'm just
All
going to quickly
yogurt
go
has
through.
This is what. Now take a look. This yogurt. Let's in the gram stain bacteria that stay bacteria. We are going to. All right. To prevent bacteria, you're going to use a striker to light the Bunsen burner. The gas is turned on low already, so bring the striker to the Bunsen burner with the flint side facing down and squeeze the striker to create sparks.
So the arrow is telling you what to do. Take your mouse and then move it down. That'll take the striker down to the
We
Bunsen
need to
burner
dilute
and
the
light it.
yogurt before we put it on the microscope. Start by placing 1 milliliter of the diluent into a test tube using. This is an inoculation. Start by sterilizing the inoculation loop in the flame of the Bunsen burner. Hold it after it has cooled down. Use the loop. Place the yogurt sample into the previously met. Now, sterilize the inoculating loop again in the flame of the Bunsen burner. Hold it Transfer a small amount of sample from the test tube to your prepared slide. Place 2 loops full from the sample onto the slide. Tilt the slide to spread the drop out slightly, about the size of a penny. Now we'll heat fix the slide. This prevents the bacteria from being washed away when we begin the staining process.
They didn't mention it, but that was allowed to air dry.
Place the slide in a clasp, such as a clothespin, and pass it over the flame of the Bunsen burner. Be careful not to put it directly in the flame. Now we begin the staining process. After the staining is complete, we can look at these slides using a microscope. We use 4 chemicals to stain a slide. We apply them in a specific order and rinse the slide off under slow running water in between each chemical. The first is crystal violet. This is our purple stain, which will indicate gram-positive bacteria. Using the dropper, squeeze out enough drops to cover your sample. Let that sit for 60 seconds. Now rinse it under slow running water. Now use the bottle of Gram's iodine. Gram's iodine will fix the dye to the cell walls of the bacteria and make it permanent. Squeeze several drops onto the sample, enough to cover. Let that sit for 60 seconds.
Anyways, this is going through the Gram stain, so do that. Are there any questions about any of this? These are the steps of the Gram stain. So you must do these steps to do the Gram stain. First, you flood the smear with crystal violet, letting it soak for 1 minute. You then rinse off the crystal violet, not rinse off, dump off the crystal violet, and then wash the slide in gently in water. The cells, both the gram-positive and the gram-negative, will stain purple after you've added the crystal violet. And this is a blue-purple color. Okay? You then add the Gram's iodine, just flooding the entire surface of the slide with Gram's iodine. And you let that sit for 1 minute. Then you dump off the iodine and rinse it in gently in water. After the Gram's iodine is added, both the gram-negative and the gram-positive cells will stain purple. But this purple color will be a little more purple than this blue-purple color. The intensity of the stain is slightly darker as well. That's not shown here, but the Gram's iodine will complex with the crystal violet, making a larger molecule than either the crystal violet or the iodine alone. Any question about any of that? All right. Then you add the decolorizer, which in this case is alcohol acetone. I will warn you that the decolorizer can be different things. Sometimes it's acetone. You don't need to know what the decolorizer is. Most of the time it's alcohol acetone, but sometimes it's alcohol acid, and sometimes it's only alcohol. After you apply the decolorizer, the crystal violet will be removed, the crystal violet iodine will be removed from the gram-negative cells, but not from the gram-positive cells if the decolorizer was applied correctly. Now it says to let it soak the decolorizer for 5 seconds and then immediately rinse with distilled water. The reason why the rinsing has to be immediate is after you remove the decolorizer, there will be trace amounts of decolorizer on the cells, and that decolorizer will continue to act. And if left long enough, the decolorizer will pull the crystal violet iodine out of the gram-positive cells. So that's why you need to rinse immediately after this step with distilled water. If you notice, I never mentioned after the previous 2 steps that the rinsing had to be immediate, because it doesn't. You could let the cells sit for a long time. I think one time I've even let the cells sit for a day and then went and rinsed the cells with water. The rinsing did not work well because the dye had dried, but it did remove some of the excess dried dye. So you do want to get the stain off the cells before the dye dries. But as long as you get it off before the dye dries, it doesn't matter when you get it off. After the colorizing step, though, it has to be done immediate to pull the decolorizer out of the cells. Now, the best way to apply the decolorizer is to watch your slide while you're adding the decolorizer. And you take the dropper and you just drop the decolorizer, meaning squeeze the dropper so the decolorizer squeezes out, and then just flood the slide with decolorizer until the crystal violets, meaning the blue color or purple color, stops running on the slide. Once the blue color stops running on the slide, you stop applying the decolorizer. And then you rinse the slide with distilled water. That's the best way to add the decolorizer. Add it while you're watching it, and then you simply add it until the blue stops running. And that will be about 5 seconds. Fourth, flood the smear with safranin. Safranin is the counter stain, and it will stain any cells that were made clear by the decolorizer into the red color or pinkish color. And therefore, after the application of the gram stain, meaning after the application of the safranin, the gram-positive cells will stain a purple color, and the gram-negative cells will stain reddish or pinkish from the safranin. Any question about any of that? After applying the safranin, you dump off the safranin, rinse it gently with water, and then you can just blot it dry with bibulous paper, and then you can view it under the microscope. All right, here we have 2 smears, both E. coli and Staphylococcus aureus, that have been gram stained. And this preparation of E. coli is much better than the above one. It's not because of the stain, it's just that the cells were better. The smear is better, the shape is better, and I'm not sure why the shape is better. And I think the picture is better too. All right, so then state the gram reaction, whether the cells were gram positive or gram negative for both E. coli and Staphylococcus aureus, and then give me the cellular morphology, their shape, meaning is E. coli a coccus or a bacillus? And is Staphylococcus aureus a coccus or a bacillus? So fill in the table here. After you've done that, then answer these questions. And this is procedure 3A. Tell me, what's the color of Staphylococcus aureus after the primary stain is added? And it tells you your 3 options are pink, purple, or colorless. And then B, E. coli, what is its color after the primary stain is added? C, what's the color of Staphylococcus aureus after the alcohol acetone decolorizing step? And then D, what's the color of E. coli after the alcohol acetone step? E, Staphylococcus aureus, what's the color of it after the counter stain is added? And F, what's the color of E. coli after the counter stain is added? Any questions about procedure 3? If not, let's move on to procedure 4. The acid-fast bacteria stain. We have a video clip on that stain. You first apply carbol fusion and all the cells will stain pinkish. You then decolorize by adding acid alcohol wash, and that will pull the carbol fusion from the acid-fast negative cells, so they will appear clear, but the acid-fast positive cells will retain the carbol fusion, and they will be red or pink. You then apply the brilliant green as the counter stain, and that will stain the acid-fast negative cells green in color, but the acid-fast positive cells retain the pinkish color. Here's the steps of doing the carbol fusion, or I should say the acid-fast stain. And then answer these questions, which is right here. It's got a question mark. Seen below are 2 smears where the organisms are denoted by arrows. Answer, is it acid-fast positive, AFB, acid-fast positive cell, or acid-fast negative? So mention that for each arrow. You can write the answer with each arrow, or to the right or below. You can say on the left image, the leftmost arrow is, and then tell me acid-fast positive or acid-fast negative. And you can say the top arrow is either acid-fast positive, acid-fast negative. That's how you answer this question, okay? Any question about that? All right, procedure 5 is looking at special stains. We have 2 video clips, one on the endospore stain and one on the negative stain to illustrate the cell capsule. And then we have 3 pictures, A, B, and C. Question A refers to picture A. Question B refers to picture B. And question C refers to picture C. So question A, and that's procedure 5, 2A. This is the Schaefer-Fulton stain. Oops. It uses what stain as the primary stain, and ultimately it dyes what cell structure that is produced by some bacteria to aid in its survival. And then A2, what are some examples of pathogenic endospore producing bacteria? And you can get these from the lab module. And then question B, this is what stain that coats long whip-like appendages which aid in motility. And then B2, state the flagella arrangement that's shown in picture B. And then B3, what are some examples of clinically significant bacteria that produce flagella? And I'll tell you, for some, you need at least 2 species. Okay, so if you only get one, you'll only get partial credit because some is more than one. Any question about that? All right. So for question C, this is what stain that uses India ink or nigrosin and can be used to detect the presence of a cell structure which is located exterior to the cell wall and prevents phagocytosis. So get that from here. And what are some pathogenic microorganisms that produce capsules? And once again, you can get that answer in the lab module. And then we have 2 case studies for this worksheet. Case study 1, a 45-year-old female is experiencing respiratory issues. Based on her clinical presentation, the doctor orders a chest x-ray shown here and a sputum for AFB culture, that's acid-fast bacteria culture. In addition to the culture, an acid-fast bacteria smear is made and stained. Based on the findings below, what is the most probable diagnosis causing the problems in the woman? You must name the species which is causing the infection. Okay. If you don't know in the x-rays of somebody's lungs look entirely dark, like this region of the lung here, and this region here and there, and this entire area over here, as well as up there, something looks wrong with the lung. And then there's the Nielsen stain, which is staining for acid-fast bacteria. All right, case study 2, a 64-year-old diabetic patient is admitted to the hospital with soft tissue necrosis, and that's shown there. And I can tell you that looks really bad. A routine culture and gram stain is ordered. The bacteria only grew anaerobically. So that's one hint. And this is the gram stain. That's another hint. And then we have the Schaefer-Fulton stain. And that's the third hint. Based on the findings below, what is the most probable diagnosis? You must name the species which is causing this infection. Any question in the lab? All right, go ahead and work in the lab. I will be here until 5 o'clock, in which case I will log off and begin the lecture. And I will be here to answer any questions. Are there any questions before I stop sharing?
I actually have a question about the procedure 2. At the end of it, it asks what the total magnification is, but there's no, like, I don't see any indication of any numbers or anything. Is that included in the video that is on there?
No. In procedure 2, it tells you which objective lens was used. And from the objective lens, you can figure out the magnification, the total magnification. You just take the power of the objective lens times the ocular lens.
Okay, thank you. I missed it.
Okay. All right, so start working on the lab. and I will be here to answer any questions.